now we're understanding that through evolution, especially in species with large brain size like ourselves, that certain processes and steps have evolved or maybe changed, partly influenced by the fact that a lot of these developmental processes have to occur over a very protracted period of time, in humans over 40 weeks. And that has impact. Now, the dogma was that by birth, most of these neurodevelopmental processes, such as proliferation and migration, had already been completed. And everything after birth was focused on fine-tuning, myelination, the wiring, and morphological changes. But again, now we're seeing that this might not be the case for everything. And why does this matter? Well, I often consider the perinatal period and kind of the black box in human neuroscience. Now, the perinatal period, that time right before and then after birth, is really from a clinical perspective, and my other hat is a neurologist, because we know that this period is really a high time of vulnerability, exposures to insults like hypoxia, infections, the propensity and potential for prematurity, really is fundamentally impactful to the development of the normal brain, and now highly has been associated with outcomes such as the emergence of conditions like autism spectrum disorder, epilepsy, and even neuropsychiatric conditions such as anxiety and intellectual delay. So we really need to understand what is happening in this black box to really then find what are the risk factors for these outcomes. Now, I entered this foray back when I was in my mentor's lab, the Alvarez Bouya lab, and I was working with a very talented student, now currently a psychiatry resident at Stanford, David James, and I forgave him for going to Stanford. We worked together and started to analyze neonatal brains. Here I show you one at 39 gestational week that we analyzed through sections taken from a longitudinal or sagittal perspective. What David and I noted was that there was this high cell density organized around this cavity, the lateral ventricle, that used to be the neurogenic niche. Now, these cells around express this marker called double cortin, or DCX. Now, this is a microtubule-associated protein that's fundamental for processes such as migration and often is used in the field as a molecular marker for young migratory neurons. So David and I were intrigued by the fact of how abundant these cells were still at 39 gestational weeks around the ventricle and the wall, but how they organized very distinctly, kind of formed the cytoarchitectural layering as they went away from the wall and started to enter the developing cortical plate. Close up here shows they went from a tangential longitudinal organization to clustering around blood vessels, to ultimately then organizing into these large neuronal clusters that then oriented and entered the overlying cortex. I won't go into the details today, but David and I showed that this was one among several populations that still were persistent through several months, up to even 7 months, in the infant brain. Previous work in my mentor's lab had showed that there were these restricted populations, the rostral migratory stream and the medial migratory stream, targeting areas such as the olfactory bulb and a small region of the prefrontal cortex, and then this expansive arc emanating from the ventricle wall, going to the overlying regions in the frontal lobe and cingulate cortex. And again, we want to highlight that what we identified with this is this unique organization, the cytoarchitectural tier formation from which these cells emerged. David and I also showed that these cells, or at least the majority, were inhibitory in nature. So with this concept of there's still ongoing neuronal migration and beyond in the infant brain, these cells were also targeting very specific regions. Even with this kind of snapshot that we had, both in time and in perspective and neuroanatomically, we could see it wasn't just a homogeneous migration, but really targeted to areas such as the frontal lobe, cingulate gyrus, areas associated with higher cognitive function, and also from neuropath studies associated with the emergence of neuropsychiatric and neurodevelopmental conditions. What really fascinated us and raised questions for us is that this process now could be influenced by external factors, external stimuli like sensory input, as well as those perinatal insults that I talked about, hypoxia, ischemia, and potential perinatal infections. So how do we study this period? It's challenging. The human brain, as I showed you with the postmortem brain, is quite large, and we often only get access to very specific regions. And understandably and appropriately, these samples are very rare and precious, which is partly why there's so many questions that remain about this time. So we turned into looking at other model systems. And we focused on, after conversations with a friend and now collaborator, Dr. Nikki Robertson, a neonatologist at the University of Edinburgh, we decided to look at the piglet. not only because it's delicious, but because it had a lot of similarities to the human brain. These piglet brains are gyrencephalic, meaning they have the infoldings that we see. And then MRI studies have really shown that the brain size, and particularly the changes that happen in this perinatal period in the equivalent time in the piglet brain, are quite similar between these two species. Now, there's been an increasing interest in genetic and proteomic studies have really highlighted actually quite the similarities between human and piglets, especially when compared to our classic rodent models. So there's been a lot of excitement about this. But before we went full hog into this animal model, we wanted to step back and think more about the evolutionary aspect of this arc structure and these late waves of migration that were taking place. And I'm going to highlight that this was work that was spearheaded by Jae-young, a postdoc, and Caitlin, a grad student in the lab, who really took on this challenge and weren't afraid to tackle a new model system. Now, again, we first wanted to ask, what is the nature, how conserved is this arc structure across different developing brains? And here I show you representations, 3D MRI brain reconstructions of the early postnatal human brain, piglet brain, non-human primate marmoset brain, and mouse brain. And I hope what jumps out to you is just the comparison of the very gyrated human and piglet brain compared to the other models, the mouse brain. You know, we were excited to have a non-human primate model as a comparative. So we started to look at the cytoarchitecture of this equivalent area around the ventricle wall. Coronal sections from this region showed that indeed the human arc that we see here now at term was present in postnatal day 0 piglet brain. It was actually absent, even though we saw cell densities at the wall in the P0 mouse brain, those emanating tiered structure was absent in the mouse. And then interestingly, it was absent in the marmoset brain. And it reminded us that while being a non-human primate, the marmoset was actually lissencephalic. Now, this arc structure should be kind of characterized by the enrichment of those young migratory neurons. And indeed, this wall area, both in the human and the piglet, had this layered organization of DCX positive cells, here shown in green, also labeled by another neuronal marker called PSA-NCAM. And these were highly enriched in the human and piglet. But even though the walls of the mouse and the marmoset contained these young migratory neurons, we didn't see this layered organization or extension out into the developing white matter. Now, Jae-young quantified these populations in two different ways. One, she generated what she called an arc area ratio, where she measured the density in the area of these cells at the wall in this extension, and then normalized it to the total cortical area, to kind of take into account brain size. She also then generated a gyrification index. This is something used in the MRI field as a way to measure the levels of infolding and often used across ages and across species. So first of all, Jae-young showed that indeed the human and the piglet have a high arc area ratio compared to the marmoset and the mouse. We were lucky that we were able to access also macaque data, a gyrated non-human primate, and this also had a high arc ratio. Then Jae-young correlated the arc ratio to the gyrification index, the GI, and saw that there was a very tight correlation between the presence of high GI and this arc, suggesting a relationship of the presence of this kind of scaffolding of late waves of neurons in gyrated brains. So we decided to pursue the piglet brain and take advantage of the fact that we could actually do whole hemispheric analyses. So I'll just highlight here, this is a P0 brain, now looking again longitudinally or sagittal. We could identify those streams that we had seen in the human, the overlying arc, the structure going to the olfactory bulb, the RMS, and we even identified the medial migratory stream, that branch going to the prefrontal cortex equivalent that we hadn't seen in other species before. But what also intrigued Jae-young and I was the fact that when we moved posteriorly, we could still see enrichment and an abundance of these double cortin positive cells at C-to-Hau outlets. So then we turned and did coronal plane exhaustive serial section analysis to really look at the entirety of all these late waves of young neurons migrating. And again, identifying and using the arc as our kind of milepost, we could see that not only did the arc have extensions and contributions to young migratory neurons in the frontal lobe areas, but also to more areas more ventral and lateral, areas like the piriform cortex and the equivalent of the temporal lobe in the piglet brain. We went back to our other species, and indeed in the human, This allowed us to move more posteriorly and see that the arc still was present in these posterior equivalent sections and also had these ventral outlets that now a subgroup of us are investigating. And furthermore, as expected now, the marmoset was lacking in these expansive streams, even though we saw a few rare young migratory neurons kind of just overlaying in the developing white matter. Now, we really wanted to confirm that there was indeed active migration taking place both dorsally and ventrally from this arc area in the postnatal brain. So Jae-young teamed up with a very talented student, Emma Horton, to do time-lapse microscopy, where they generated organotypic slice cultures from the P0 piglet brain and infected the arc area with the CMV-GFP adenovirus. Here I show you a culture at day 1 where you could see the labeling of the arc cells. And I'll play this video again, taken on the left from dorsally migrating cells and from the right ventrally migrating cells. And I hope you can see, kind of just off the bat, the difference in their organization and their behaviors. These cells seem to be kind of more individualistic, going out longitudinally or radially, as opposed to these slower moving, what appear to be slower moving cells, organizing in clusters. Now, Emma did post-hoc staining to confirm that these indeed were these double Cortin positive cells that we were studying statically in our sections. Now, given these differences, Emma quantified the maximum, minimum, and mean speeds, average speeds of cells from the ventral and dorsal streams. And she was a little bit bummed when she saw there wasn't really any statistical difference. But she astutely noted that in the ventral streams, there was this subpopulation, a small proportion, actually not that small proportion, that really appeared to be still relative to the rest of the cells and in comparison to the dorsal population. I think this is better seen in our histogram of the frequency of the different speeds in these two populations, where again, this ventral population really had an abundance of kind of still cells or cells in the pause state. So taking each cell and quantifying their motion, she identified what she called 2 modes of migration, or 2 modes of behaviors. One more a migratory mode, where the vast majority of the time, over 70% of the time, was spent at speeds over 3 microns per second. And then what she called a more resting mode, a mode 2, where cells were more in a resting state, really either still or traveling less than 3 microns per second. And to cut to the chase, when she compared the dorsal and the ventral cells, she saw that the ventral cells were actually, the vast majority of them were in this second mode, really in a pause state. So it wasn't that necessarily, if you look at each cell, they might be quite equivalent, but as a population, they really had different behaviors. And so this raises a question for us, what are the differences? We knew from our human work, and now we corroborated it in the human and piglet, that these cells were inhibitory neurons in general. But the field knows that inhibitory neurons are quite diverse in origin, and we think in their function. So do these differences in the type of inhibitory neurons underlie these differences in behavior and their targets? So this diversity of inhibitory neurons, I'll just say, stems from the fact of their embryonic origins. So here I show you an embryonic mouse brain, where we look at this ventral region, ganglionic eminences, as they're called, because of the little neurotamic bumps that you see. And these are really the sites of all the diversity of cortical inhibitory neurons. And not only can they be identified neuroanatomically, but each of these subregions express a different profile of transcription factors that helps also differentiate these regions, separating them into a lateral, medial, and caudal ganglionic eminences. And why is that important? Not only because they're anatomically different, but each of these subregions generates a different profile of inhibitory neurons, which is really fundamental. And we don't even have full knowledge of what these differences are between these different subtypes of inhibitory neurons. Now, the vast majority of attention has been to the MGE and CGE populations because they make up the majority of cortical inhibitory neurons, and in particular, the MGE or medial population, because in the mouse, they make up over 2/3 of the total inhibitory neuron population. Though I'll just throw out there, might not be the same case in the human. So we were curious, as we were trying to differentiate and better understand the differences between these dorsal and ventral streams, what's the composition? Does that help explain a little bit some of their differences? So in the last minutes, I'm just going to highlight some of the work by another grad student in the lab, Anoy Podar, who went in and performed highplexing in situ hybridization, where he was able to look at over 30 genes either with relationship to their GABAergic in nature, so GABAergic transcription factors and other genes, or neuronal migration-related genes. And then using double Corton as a way to identify our cell or region of interest, he then started to quantify the expression profiles within each of those cells in either the dorsal or ventral streams. And here I'll show you fields of view from different parts of the stream, and I hope it just jumps out to you just qualitatively, visually, that each of these streams have a different expression profile. And again, this is looking at fields of view taken from these ventral stream populations and cells from the dorsal populations. Now, taking the analysis that Anoy did, he both generated a heat map and to identify what is the distribution of the genes and how do they break up into the different neuron subtypes. I think, first of all, this heat map just confirms the fact that the vast majority are inhibitory in nature, though one thing that we're investigating now is that there is a small subset that are excitatory. That was really surprising to us. Then he took all these cells and put them through a Seurat pipeline to identify, can he break down different even subclusters of inhibitory neurons? And I just get to the crux that, indeed, he was able to identify that excitatory population, but also 6 different inhibitory populations, 4 of them, actually 5 of them being CGE related. So there was a contribution from the other subregion of the ganglionic eminence, but nothing compared to what the CG was contributing, which was really, again, fascinating to us in terms of the contribution of this subpopulation. And then I'll just end on the fact that he could take that data now and put it back, since he had both the gene expression information, but now the spatial information, he could look at the topography of these cell clusters. And I hope what jumps out to you is the fact that if you look at the dorsal stream at 2 different AP levels and the ventral stream at 2 different AP levels, each of these streams has a different composition with respect to those clusters. The ventral stream is really kind of highly populated by a SPA-CUPTF2 population, the dorsal stream more by a VIP population. And again, each of these are a different subtype of interneuron that each have a potentially different function. So we're really trying to understand the diversity of these cells now and how they might relate to those molecular signatures of each of these streams. So I hope I left you with this concept that the arc, the structure at the ventricle wall in the early postnatal brain, is really a unique feature of what we think present in gyrencephalic brains, populated mainly by GABAergic interneurons, but TBD what the excitatory neurons are doing there. These are extensive, they contribute to extensive populations that we know go not just to frontal areas, but also now more ventrolateral areas like the temporal lobe. And surprising to us is that they have a diverse array of interneuron subtypes, but really predominantly come from a caudal or a CGE-derived origin. And each of these streams has a unique molecular signature to them. And I think that's important because, and something I won't get into today, we're now seeing even between the timing of these two streams, the dorsal one going to frontal lobe, the ventral one going to temporal lobe, that there's a temporal difference as well, where we think that the migration to the temporal cortex outlasts that, a migration into the other areas. And so we postulate that this late wave of migration is a cellular mechanism that can create regional specificity. Particularly, again, these are areas of higher cognition, and these waves could be ways to enhance or contribute to plasticity and the full functional acquisition that is taking place in higher cognitive functions in these gyrencephalic brains. But this also opens up new windows, new vulnerabilities to postnatal experience and postnatal injury. It's what I want to end on, again, thinking about this as a community, are questions that we're interested in, and maybe with some people we could team up or even just brainstorm together, thinking about what are unique features of these late waves of neurons? Are they similar to the ones that were migrating in the prenatal brain, a much simpler, less complex environment? What are the origins of these late waves of neurons? Does this suggest that there is ongoing neurogenesis? And there's data I think, from our lab and from others that says there might be late neurogenesis taking place in these larger brains. And then lastly, I think really being creative, especially now that we have a good model to actually look at personality changes, sociability changes. What are these last waves of interneurons doing with respect to behavior and function? So with that, I thank you for your attention. I want to thank all, obviously, my lab, who are just the exciting generators of ideas and the beautiful work, collaborators at UC Davis and the University of Edinburgh and in other departments at UCSF, and obviously to the UCSF Swine Center and to the patients who bring a lot to the table in thinking about these questions. Thank you.